Good morning, everyone, and welcome. We'll begin our presentation in just a couple minutes. We're giving everyone a chance to join, and we will begin shortly. Thank you. Okay, so I think we'll just begin, and I want to welcome everyone. My name is Marissa Fraser Morera, and I am with Pion and will be moderating today's event. We are very excited to present de-risking development guiding formulation strategy with Pion's MacroFlux to predict human outcomes of KinetiSol process Etravirine. And we're featuring our guest speaker, Dr. Daniel Davis. Daniel is the Director of Business Development at Austin Pharmaceuticals, or AustinPx. And they are a CDMO specializing in early phase development for the oral delivery of small molecules based in the San Francisco Bay Area. And he leads the strategic partnerships and early phase program development with biotech companies. Daniel received his bachelor's in chemistry and doctor of pharmacy in 2019 from the University of Texas at Austin, graduating with both academic and research honors. He went on to complete his Ph.D. in 2021 through the PharmD Ph.D. honors program in the Division of Molecular Pharmaceutics and Drug Delivery, and he is currently pursuing his executive MBA at the McCombs School of Business. Previously, Daniel served as a principal scientist at AustinPx, where he led multiple drug product development programs. His work focuses on enhancing the solubility and bioavailability of challenging compounds through innovative formulation approaches using KinetiSol technology, spray drying, hot melt extrusion, and selective laser sintering. He is also a co-editor of the third edition of Formulating Poorly Water-Soluble Drugs, and that is an AAPS Springer edition of 2022, and has co-authored over 30 peer-reviewed publications, abstracts, and book chapters. He is also a co-inventor of 10 patent applications and formerly served as social media editor for AAPS. We really appreciate Daniel taking the time out of his busy schedule to join us, and we are really thrilled to have him here to share his expertise and insights. Also joining us today is Balint Cinco. Balint is our VP of Innovations at Pion Inc., and he is going to be assisting during our Q&A for all flux-related questions or Pion product questions. And then just briefly before we get started, I just want to quickly go over a few housekeeping details. If you need technical assistance during the webinar, please send your query to me via the chat function. You are encouraged to contribute to the success of this webinar by submitting questions at any time during the Q&A. And there is a widget on your screen for the Q&A and one for the chat, so they're separate. So send your questions on the Q&A widget. Our speaker will answer the questions at the end of the presentation. And any questions that we don't address today will be answered offline after the event. Lastly, a recorded version of this webinar will be available on our website within about 24 hours. And you will all receive an email with a link to the recording that will be sent to all registrants. So now, without further ado, I think I will hand things over to Daniel if you're ready.
Yep. Thanks, Marissa.
Great, thank you.
Appreciate the introduction and the opportunity to talk today at these webinars. I've watched a lot that y'all posted over the years and excited to be a part of this. And so to give everyone a little bit of context, we'll go introduction, give a little bit more about Austin Pharmaceutics. I know Marissa told us about it, at a high level, but just give a little plug to that. Some background on like ASDs, pretty basic, just to kind of set the context, tell you about KinetiSol, just enough to understand kind of the angle and the position that we took as a strategic way to enter and go about generating a generic formulation of the case study that we're going to be talking today, Intelence, and really just trying to show how dissolution and fiber optics can be such a strategic tool when used appropriately, and how it can be, misleading, and you really need to be coupling in vivo data as you get in vivo data to update your in vitro method so that your discriminating method is also predictive at the same time. And so we'll guide you kind of through a playbook of what we did for a BCS class 4 product using human data to ultimately create a generic equivalent Intelence product that was made by spray drying. So a little background about our company, and this will kind of correlate to the spray dry product development. We started as a technology company solely centered around KinetiSol processing, which is basically just a different way of manufacturing amorphous solid dispersions compared to the traditional hot metal extrusion and spray drying technologies. That started in 2007, 2008 period. We spent the past, 15 rough years better understanding that, preparing for commercial, developing our own products as a sponsor company. We took 6 programs through clinical trials, 5 of which were 505(b)(2)s, and one of which, the case study today, was a generic product. In 2022, we rebranded from the sponsor side to more of a CDMO, where we're fee for service, and we really take the expertise that we've learned of being world experts in amorphous solid dispersions, in oral solid development for small molecules, to giving that to our clients. And so now we're solely fee for service based, taking an agnostic approach, not forcing KinetiSol by any means, but really understanding what your molecule needs as it's coming out of discovery and as information comes available to pair the best formulation technology, considering your clinical pathway and material limitations for that molecule. And so this is just a rough, developability classification system that kind of guides these approaches. If you have a high solubility, high permeability compound, and maybe it's a low dose, we can move forward with a simple capsule formulation. But as you get into these cases, as we'll talk about today, where some more complex formulations may be required, you also know that you're placing yourself with someone that has the ability to do all formulation developments. And so we're more than capable with enabled formulations, lipid-based, spray drying. We can move your product through spray drying all the way from discovery with low amounts all the way to a phase 1 product in that end. But additionally, we have the KinetiSol processing in-house to assess that. And so looking at these kind of class 2B, class 4 compounds where some solubility enhancement may be required, that kind of brings us into what is an amorphous solid dispersion. And not going into the full conversation, but a really basic high-level overview of this is, you have a crystalline drug that is very tightly bound crystalline lattice, and we're disrupting that crystalline lattice and creating this meta-stable state, with some of the benefits of that being the lower activation energy to create a supersaturated state and increase bioavailability. And so again, another kind of crude drawing here to represent a very complex topic, but basically picture the yellow circles here as discrete APIs, or amorphous drug, and then the squiggly lines as a polymer or an excipient or a stabilizer, however you want to frame it. That's basically allowing this metastable amorphous drug to be stabilized in a state that is acceptable for pharmaceutical use. And in addition to physical stabilization, there's a consideration of this stabilization of performance that needs to take place in vitro, where most, this is an adaptation now from 15, 20 years ago, where we have a spring, the amorphous drug springs in, and really your goal is to maximize this curve. There's been some more publications that have come out where this curve is, the goal is really to keep this as high as you can for as long as you can to maximize your AUC. So this is kind of the background and approach of why you use an ASD. How do you stabilize it to, de-risk that physical instability, but the benefits that it can have over a crystalline drug. And how relevant are ASDs today? This wasn't a concept of even considering these in the market before the early 2000s. It really wasn't until 2007, 2008, where we see the first ASD products introduced into the market that kind of kickstarted the adoption and showcased how powerful um, an enabled formulation can be for a product. And so we see in 2008, Intelence being the first product by Janssen for to be brought to market for a very challenging ECS class 4 drug. But before this, there's a lot of work that went in the backgrounds to get this technology to where it is today, where spray drying and HME are the two, recognized manufacturing strategies for all ASDs on the market today. And that started in the 80s and the 90s when these technologies were adapted from another industry and spent 20 years incubating in the academic setting where, my PI, some may know Jim McGinnity or Bill Williams or even Fong Zhang, who've been working and bringing a lot of this information to de-risk this and increase our understanding of what when an ASD is appropriate and what the benefits it can have of that. And I'll speak about that more specifically to Intelence to kind of emphasize that benefit that was seen. And to introduce KinetiSol into this and how it plays into this case study, I wanted to be able to show you this diagram here. And so we feel KinetiSol, is a fusion-based technology similar to hot melt extrusion. However, it mixes at a higher mixing rate, shorter processing times, doesn't require the process to be at the melting point of the API, and uses lower temperatures. And so we feel that anything you can do by hot melt extrusion, we can do by KinetiSol. And then similarly for spray drying, we showed this larger design space due to KinetiSol not requiring common solvent miscibility, or even just your excipients having to dissolve in your solvents. And so we're able to process basically anything that fits for your molecule. And so the thought process here is we use a technology that can process anything to be able to make the best formulation for the drug versus using a technology that limits the design space for your drug by that technology. So showing how KinetiSol works, and then we'll dive into the case study, is think ultra high shear granulation, just very, very quick mixing. There's hopefully the video is showing for everybody on here. It really helps illustrate this. But quickly on how this is working, we have these mixing elements within a chamber as they're getting up to speed at very high speed, think, like 5,000, 7,000 RPMs, forces all the material to the exterior wall. And that material is forced to pass through the specific gap width. And that gap clearance causes friction both between the drug particles themselves and the chamber wall. And that friction alone is what causes an increase in temperature and a phase transition. And so that powder, polymer or physical blend transitions into a molten mass. And that molten mass, think about it as kind of a surrogate as a solvent almost. It's a viscous solvent where your drug is dissolving into that molten mass. And your drug is converting from a crystalline to the amorphous phase solely through a dissolution-based process. That's going to be respective of the mixing speed, which is incredibly high in this case, and the processing temperature. We're able to monitor this in real time using an infrared probe that takes measurements every 0.1 seconds. And that's kind of what the process is ended on. And you get this molten mass that is then quenched and milled and used for downstream testing. And we'll kind of talk to some of those attributes that impact our dissolution by some of the pion measurements later on. Kind of touched on this, really just showing the dissolution process. This is what allows us to create single phase ASDs within our process. And now switching more, so that was a background to kind of set the stage and why, KinetiSol is being used even in this case study, and it was even considered as a strategy that we were taking to be able to create a generic formulation of Intelence today. And so This Intelence or Etravirine, as I'll refer to it moving forward, is a non-nucleoside reverse transcriptase inhibitor for HIV treatment. This was, one of the first products brought to market for this. There was a really high clinical need. This was before Gilead entered the space. Also, a lot of spray-dried products for this indication as well. Intelence specifically was a BCS class 4 rapid recrystallizer, insoluble in water and acid. But there's a really high clinical need. And so innovation typically follows that and that clinical demand in the market and being able to help patients of unmet need here. And so when this was being dosed in the clinic initially, I think it was most likely for their phase 1, but it was 36 tablets per day was being used for this early development work. And so you can imagine the pill burden alone, the doses that were required. And so Janssen, was an early adopter of spray drying. They built the expertise in-house. They built their PST4s. They've, now are building for commercial for their own products across the board and are world experts in spray drying. And so this was a very complicated product for them to bring forward at the time, high risk, but the market was there. And, 36 tablets is, unacceptable, but getting that down to one tablet really starts to show the value of what an ASD did. And so there was a case to bring forward for the rest of the pharma on the market at the time. This was it. It was like, look, this is the power of what you can do by bringing an ASD to a formulation, increasing your bioavailability. You're going from 36 tablets to one. And so that kind of sets the stage of this. This was a $500 million product per year as it was approaching the end of its patent life. It's a major generic manufacturer actually failed to match Intelence as they were approaching to bring this to market. And that was because they went to an approach that wasn't spray drying. The infrastructure wasn't built yet for them to be able to go and get this spray dried cost effectively. And so this is kind of where KinetiSol comes in. Again, I mentioned this was the earliest product, so this is why this is coming off patent first. But we felt KinetiSol comes in here because we have a lower manufacturing cost, and we're able to make challenging formulations that can't be made by hot metal extrusion in some cases. And this is a great example of that because the Etravirine product actually, I think the melt's 260, but it actually starts to degrade at like 50 degrees below melt. So like 210, you start seeing some degradation. So this makes this very challenging to manufacture by, or basically impossible on hot metal extrusion, or very challenging. I won't say impossible. And so spray drying was kind of picked as the product for them to move this forward. And so to set the stage of some of the challenges that we faced when developing this is, as I mentioned, BCS class 4, low solubility, low permeability. This causes challenges when developing dissolution methods and trying to correlate some of that data. We're also using a fusion-based technology versus a solvent-based technology. And so we have different particles that we're trying to match. And so we have this spray dried comparator that, probably making less than 60 micron particles. They're hollow spheres. They result in rapid dissolution compared to a more dense fusion-based approach where we have to select and we can pick our particle size range and we balance that with dissolution speed and tabletability. And then lastly, as a generic application, the label actually stated that it's required to take Fed, but to be able to move a product like this forward, you actually have to match both the Fed and the fasted state, especially in the EU for these products. And so some project goals before we dive into some of the data was, you need some way to test these in vitro and to discriminate. And the goal, initially is just, can we discriminate between formulations to put our best foot forward into some, cheaper preclinical models, in vivo studies before we test this in humans, and then just keep iterating. And it's a cycle, and we get better each time. And with the ultimate goal of establishing bioequivalence in both the fed and fasted states. And then upon that, the success of that, file a generic drug application with the FDA and in other markets. And so this is kind of just our compass here. This is our goals of the product, was to develop both fed and fasted matching. One thing to note here is the big discrepancy between fed and fasted. that you see, and so it should be required to be taken with food, but also having the issue with having to match with fasted when you see two completely different PK profiles, and food effect being, 6x here between the fed and the fasted state. So I thought it would be kind of relevant and really to just make a point and emphasize here by starting with some QC dissolution media and trying to set the frame and the context of going back to, this product was brought to market in '08. Let's go back to when we were doing this in starting this in like 2015 to 2017 time range. We're increasing understanding of ASDs, of what we consider biorelevant dissolution media testing. I'm starting with the QC dissolution media. You'll see at the bottom, it's a pH shift. It's finishing with 900 mLs of 2.25% SLS. And so you'll see with this incredibly poorly insoluble drug, we're seeing, I didn't update the labels. This should say percent release on the side, sorry. But basically we're approaching 100% release for both the spray dried and a KinetiSol product at 60 minutes. And this is great. And we see maybe what could be some significant differentiation between these products here in this case. And so we can go and keep developing different formulations around this dissolution product and use that as a rank ordering technique as a place to start. But you want to gut check that in some capacity. And this is just to show, though they look the same and at 1 hour they have the same endpoint, and you would expect as you keep going, likely at async conditions, these would stay the same. And so your differentiation is coming here. It's slightly differentiating potentially, but when you test this in vivo, you see almost a flip between what was looking the best here and there. And so this is really emphasizing your dissolution rate in a sync condition versus maybe what supersaturation is going to be differentiating. So you're kind of differentiating between two different targets in your target product profile here. And you need to be keeping that in mind when you have a BCS class 1, where maybe this is relevant versus a BCS class 2 or a BCS class 4. And I think this is just the same media, just showing a different formulation. The red is the spray dried product, and all the others are the KinetiSol formulations. And this is clear that this product isn't releasing nearly as fast. Dissolution rate's slower. The endpoint isn't there yet at 60 minutes. But when we switch this to the in vivo study, where frankly is more important, we're seeing a flip flop here, and that the QC method that we were using underestimates the exposure in the dog compared to the Intelence product. And at the end of the day, this is slightly discriminative. This is what you want to see. You want to see discrimination between different formulations. I didn't put the exact compositions of these on here just to simplify things, but we start to see that this method can differentiate slightly. It's just maybe it's differentiating something that's not really relevant for this product here. It's more looking at a dissolution rate than we're looking at an actual supersaturation state. And so, taking a step forward here, the next step is, okay, let's look at a biorelevant dissolution media test to be able to differentiate our product. And, the understanding, there's a lot of publications out today. A lot of good groups have looked at this and different strategies. And, even as a CDMO, we try to, you bucket things, right? And you learn and you see what's best for what type of drug, like a weekly basic compound versus just a traditional BCS class 2 versus a BCS class 4. What do you do for your dissolution media? And I think, there are better best practices established at this point. But at the time, we just went with the simple, this is even for FACID V2, just 500 mLs FACID, pH 6.5 as a biorelevant approach. And, looking at this data using the fiber optics here, we're happy. We're seeing differentiation in our product. And this would, without any PK data, this would be kind of your guiding compass. It's like, okay, this is biorelevant. We're seeing discrimination. Now we need to make sure this formulation, 47? How do we improve this to make it match this? And I think the point that I'm starting to drive home is that, just because you have a differentiating method doesn't mean it's predictive and as biorelevant as you want. And so we want to make sure we're starting to see this in vitro tool being predictive of at least a rank ordering perspective to be able to gut check things. This is, at this point, this in vitro data that we have here is misleading. And one aspect that we have to check as formulation scientists developing using Pion is to gut check this to make sure what we're measuring is also accurate. And so what we can do is, do manual pulls to confirm some of the HPLC data that we're seeing. And the goal here was just to use, an HPLC method with a few different filter sizes to distinguish, what we're seeing by HPLC, and by Pion, the fiber optic is confirmed. And, we probably could have done a little better on these later pools here, but the point gets across in the first 2 hours showing that we see a high overlap and really a gut confirmation of what we're measuring on the fiber optic is accurate at this point, and that there wasn't any shifting. We're confirming some of our data. And what we know now about the colloids in this space, and that we'll see in the acceptor data, is we probably should have done some ultracentrifugation here, knowing that some colloidal species were forming to try to identify free drug, if that would have been more powerful. However, the Flux tool is a great accessory to be able to distinguish that out without having to do ultracentrifugation every single time for your product. And so knowing that our fiber optic data was accurate, we continue to move forward with, as I mentioned, maybe creating a more discriminative HPLC method, or I'm sorry, dissolution method for our product. And so I wanted to show this figure here of, I like to think of the acceptor data kind of as my gold standard most of the time during development, especially early when I'm not seeing any in vivo preclinical data yet that we've generated. And so it's a great way to decrease some of the noise or maybe variability between your samples and really get some great consistent data across the board. And so here, as you will see, the formulation that we took later, that you'll see that we took into the clinic, using these dissolution conditions down here below with the pH shift now and 900 mLs of FASIV, we see this huge discrimination between our two formulations here and the acceptor compartment when we're using the flux. And I guess I should have, to rewind, probably should have gave a figure of the flux compartment for Pion, but really you have your traditional donor set up with your, just as you would do a normal USP type 2 dissolution, and you add this second, what we call acceptor compartment that uses a pampa membrane, and you apply this, lipid membrane on the front of it, and then passive permeation is kind of measured across that membrane where you have a fiber optic probe and what we call the acceptor compartment. And so you're collecting two types of data, your traditional donor data and your acceptor data that's permeated across this membrane here. And so this data is showing us, a huge discrimination. But when we test this actually in the dog model, we're actually seeing the most similar that we've seen across the board in this case. And so again, another example of really great use of flux data. There's low variability here. We see some discrimination, but this is just telling us that though we're seeing discrimination, this isn't a predictive method. And so we have to go back to the drawing board and modify our dissolution data, our dissolution method a little bit more to ensure that we're able to basically change our testing conditions to be able to get some in vitro tool to be able to match in vivo data at the end of the day. And so I overlaid this here. I think it's nice to be able to see both the donor and the acceptor profiles. Here we changed the media again and the conditions slightly. We did 900 mLs of FASIV, pH 6.5, 50 RPMs, and we crushed the tablets. It's a little story for a different time on why we crushed the tablets. We saw different disintegration rates. And we're really trying to optimize at this point, our intermediate being matching the intermediate and trying to deconvolute these. So we crushed the tablets in this case, and we see that even in the donor compartment still, where the Cmax for the KinetiSol material is lower than Intelence, but due to that lower supersaturation, we have a lower precipitation rate potentially. And so the spray dried material goes higher, but also achieves a lower low. But when we look at this by the acceptor compartment, which is what the flux is measuring here, the permeation, and kind of deconvoluting some of maybe the secondary colloid species that are present in the media, that actually represents free drug available for permeation. We're actually seeing a really nice, one of the better overlays, especially at this 10-hour time point, of being able to say maybe these formulations are performing pretty similar. And this circles back to that dog PK that we had before. And so now this didn't change, but we started changing our method, our dissolution conditions to be able to tailor to this data. And so with that, we kind of started to see the point of like, well, maybe we don't use the donor data in this case for this exact project, and the acceptor data is more valuable. And so moving forward, we use a lot of the acceptor data to be able to guide formulation decisions. And I'll kind of show how we retrospectively did this. At the time when we were developing this, we actually didn't have a flux, the MacroFlux or any flux equipment from Pion. We only had the traditional kind of UDIS system where we're measuring donor compartments. And so this shows the value that we could have had if we would have had the Pion flux from the beginning, just trying to correlate with some of the dog data versus having to move forward some of this development on our end to human PKs with just some of the dissolution data that we had early on. And so I'll show a slide here. We actually did a pretty good job, I feel like, matching without having all these tools on the front end. So we matched the fed and we missed on the fasted. And so this kind of shows after the fact when we get when I showed this PK data here, what showed bioequivalent for our human PK for the KinetiSol formulation versus the Intelence. When we back ran this with some of the acceptor using the flux tool that we had to characterize those slightly different conditions here, but very similar to what we did, the FACID pH 6.5 versus FACID pH 5. We see the high correlation of the fed state for both the KinetiSol and the Intelence formulations. However, when we use the same conditions that we showed previously of what we considered a biorelevant predictive test for using the flux component, we see this large deviation here between the product we tested in the clinic and the Intelence product that we see here. And so a simple test of being able to run this before we went to in the clinic, we would have been able to see a very similar outcome in vitro for what we saw in vivo. And so, as I mentioned, hindsight on this, we purchased, we met Pion, we learned about the capabilities of some of the Flux tools, and then we purchased this after the fact. And this is kind of our North Star for development afterwards. But we kind of show, I made a table here of how you can roughly show, just like even without even doing a lot of complicated analysis, if you're looking at the exposure, from AUC 0 to last or 0 to infinity in humans, you're somewhere between 71 and 76% of the total exposure of the Intelence to the KinetiSol product. And then when we look at the flux amount, if you just pick a point, there's a single point at 5 hours versus 10 hours, you see that we're following really within that same equivalence that we were seeing between the humans. And then just to take it a step further, to allow us another tool when we're looking at fasted dogs, the AUC between 0 to 12 hours also kind of lines up in here. And so if we segregate this table from, to the right of this data over, we're able to see how we can have some in vitro tools that may be predictive of what we're seeing in humans, and also some like preclinical in vivo tools as well to kind of gut check our flux data that we're generating through this. And so you only have so many attempts as you're going into human studies, just to how costly they are with this. And so what we did was we need to reformulate, and though we match fed, and that's how the label states to be taken, we also need to match in the fasted state. And so we spent some time thinking about what's the minimal changes we could be making to this product to be able to, maintain our fed profile, but also match on our fasted state. And so we identified 3 parameters here of basically now what we wanted to do was that we have what we considered a validated model for in vitro testing to correlate to some in vivo data, we wanted to modify the formulation or some of the processes. And so some things we looked at was milling speed, which is a surrogate for particle size, the lubricant or the solubilization agent that we were using. In this case, it was SSF, and then also the injection temperature. And so we limited it just to 2 temperatures just to really discriminate and show a difference between that. And so the first variable, looking at what I would consider particle size, again, lower RPMs, slightly larger particle size, higher RPMs, smaller particle size. And we see when we have 0.5% SSF at a constant injection temperature, so basically these two constant different RPMs, we see the flux is highly overlaid. Happy with that data, really not expecting a large impact here from particle size. Similarly, when we look at the 1% SSF at a fixed temperature, again, we see that probably the particle size isn't the big formulation lever that we're trying to pull here to see us increasing our exposure to match that of the Intelence product in the fasted state. And so the next thing, we're going to keep the milling speed the same, so 15,000 RPMs here, and we're going to keep the amount of SSF the same, but we're going to change the ejection temperature. And we see this is, this is overlaid highly. We might see slightly higher variability at the lower ejection temperature, but for the most part, like this is an agreement across the board. However, when we look at the 1% SSF and we start looking at the differences in temperature, we see a difference here. Maybe it's not full amorphous conversion, but most likely it's, there's some level of interaction that SSS is actually having with this composition, and it's exaggerated by the temperature here. And we see, differences at different ejection temperatures that we didn't see. And then additionally, we kind of start to see, this is achieving around 14 micrograms per ml at the 0.5% SSF, whereas at the 1%, we're around 12 in this case. And I think the next slide here really teases that out. When we keep ejection temperature constant, we keep milling speed constant, and we solely look at the differences in SSF here, we're seeing that there's an increasing the SSF concentration has an inverse effect on the flux. And the formulation we took into the clinic previously was a 1% SSF formulation. And so just by slightly changing the amount from 1% to 0.5% SSF, we're seeing this huge boost. Huge is relative, but the method here is very sensitive to changes in the concentration because it's such a low fluxing compound, BCS class 4 molecule. And we're seeing a superior acceptor concentration profile curve here for the 0.5% SSF. And so what we did is we overlaid this with the Intelence product that we had in the previous formulation that we ran in the clinic. And I put some labels here just to make it very simple. But the product we ran in the clinic using, all testing conditions the same as what we previously did, achieved, roughly 10 micrograms per ml. But when we go to this 0.5% SSF here, using solely just the in vivo tools that we iterated over time to tailor both to the PK data and the human data that we got, allows us, at least in vitro at this point, to say we feel pretty confident. We made some formulation changes, very minor to the composition, but they have a major impact on the outcome, and to give us some confidence as we go to, spend the millions of dollars to run another healthy human study, basically to show bioequivalence, that we're feeling really confident going into this. And so we really leaned on the Flux tools here. And you'll notice that we don't show any donor data throughout this process because there was an alignment and there was an agreement, and it was something we kept chasing. And as we modified stuff, and I didn't capture everything we did here, but we time and time again, we came back to the acceptor data here to really guide formulation decisions. And it's not all cases, but we're going to have this much information, and you have to put your best foot forward with what you think is going to be biorelevant at the time, and then you generate this data and you go through this. And in most cases, the goal is to maximize your exposure, and you're not trying to fit a specific target product profile like this. At least when you're, have a really high pill burden in this case. So to refresh kind of the purpose here, the project goal was, to match the two. And when we went back and we dosed the modified formulation in vitro, we were really happy that we were able to achieve, a bioequivalent product in the fasted state by making these modifications that we did using the Pion tool for development. And so, that's just one of probably 50 examples we could give on this, with how much flux that we run in Pion dissolution testing that we do at Austin Pharmaceutics now. I think we're, in addition to being experts in ASTs, we really rely heavily on the fiber optic equipment that they have. The updated software has been incredibly powerful for us to do even more complicated predictions using, some of their prediction software with that. But yeah, the flux tools, we have the MacroFlux, the MacroFlux. I think we have 3 MacroFluxes now and 2 MacroFluxes. So we really leverage this as early as we can in development, whether the dissolution method we're using is misleading or not is a different story, but we're starting to generate that data. We're getting we at least can see the compounds fluxing, right? As you start getting these high molecular weight PROTACs or just beyond the rule of 5 compounds through here, you can get a feel for your compound and just get more data. And so this was really a tool that helped us for a BCS class 4 drug that was forming colloids in solution, had low solubility. And yeah, this this BCS classification that we showed is kind of a rough tool guide of how we go through development with that. And so just acknowledging a few of my colleagues, Sandra and Doran put in a lot of lab work into this before I even joined Austin Pharmaceutics, and then also Dave Miller and Bulent, as always, for looking at a lot of our data and kind of brainstorming some ideas with him throughout this process. So with that, we can switch to questions. And just thank everyone for attending today.
Thank you very much, Daniel. We'll move on to the Q&A, if that's okay. There are a few questions here. First one, is it possible to use MacroFlux to evaluate food effect for pool-soluble compounds?
Yeah, and I can, I didn't emphasize on that too much, but I had that data here in this slide. We did use FESIV conditions in our donor compartment. I will say FESIV can be challenging in the donor compartment to kind of monitor due to all the material and the species that you're incorporating in there. But Acceptor is really able to allow us to kind of differentiate a lot of that data in here. And we did see, even without having developed a fancy method, the Acceptor was able to show agreement with some of the human PK data that we had.
Okay, thank you. Next question. Can you do 2-stage dissolution with MacroFlux? And do you see any value in that versus your discriminative tests? I know this is something that has been done with MicroFlux, but not sure about MacroFlux. Thank you.
Yeah, we basically, as an organization, always start with the 2-phase dissolution approach with MacroFlux. pH shift typically like pH 2 using SGF in some version for 30 minutes typically, and then we transition to FASIV V2 pH 6.5 typically for this. And so yeah, the MacroFlux is set up where you can do that as long as you have enough of your drug product at that time.
Okay, thank you. Next question. Was your acceptor compartment the typical HEPES buffer?
Yeah, we use the default Pion supplies with them for their kind of the sync component, as you would call it.
Okay, next question. Great talk. Thanks for sharing your experience and data. Why do you run the acceptor compartment to 10 hours? Does this reflect the limited surface area or volume ratio for permeation?
It's a good question. It's two things. Sometimes as a scientist, you just love having data, even if it's not relevant. And since it's fiber optic, you're not actually doing the work. And so if you set this up in the evening, we just let it run. And it's like you do what you want with the data. And sometimes you would rather have more data than less data. In this case, this table, I think, shows we could have justified stopping this at 5 hours or a time point sooner. Right now in development, we typically do 4 or 6 hours. 10 hours, I will say, is a little overkill in this, but it depends on what you're seeing for how long your drug's being absorbed, where it's being absorbed in the smaller intestine, and you can set that. But yeah, we don't ever, especially for immediate release, not going over 6 hours typically.
Okay, thank you. Next question. I have been using Rainbow for over a decade, and I have zero doubts the data it collects. Lately, I keep running into people I check with HPLC, but I really don't think folks should be doing that as it comes from some kind of institutional doubt. Am I being too naive to get frustrated when I witness these doubt, which I perceive as emotional and not scientific?
I think it probably comes from two things of some experiment happening where there was some level of speciation, I feel like, where there's a discrepancy between what may be free drug, and it's a way to gut check that. We, as a CDMO now, we see a lot more people with this same question. And for us, we need to do what makes the customer wants in this case. And so we do it a lot just to confirm as a gut check when we do our first run. And so we'll do an HPLC pool, even if it's at just like 1 or 2 time points, to show that the data that we're collecting is relevant. But the point you make is good, because if there is a lot of speciation, you could ultracentrifuge, you could centrifuge, and you could filter, and you may see 3 different values. And it's really what's relevant for your product. And the acceptor data will also help deconvolute that as well. So I get the frustration because it's extra work and you have this great tool, but there is a level of, you can justify it later confidently if you see that overlap. And if you do see a discrepancy, it just gives you something to keep in mind.
Thank you. Let me see. Uh, Great talk. What is the best dissol system for oil-based long-acting injectables? What buffer?
Oh man, Blunt, I may let you, do you have on that one? I don't do a lot of long-acting injectables, so I don't have a lot of experience there, Marissa.
Yeah, I think soon we will have hopefully a webinar that will talk about long-acting injectables and what to do with those, and requiring a special sample holder. So yeah, I think in terms of MacroFlux, yeah, it's probably less relevant.
Okay, thank you.
Next question. Can we estimate the impact of PGP inhibitors using MacroFlux?
I'm not aware of that capability. Blunt, maybe you can correct me if I'm wrong. Since we're just using kind of a Pampa membrane and looking at passive diffusion through this kind of lipid barrier that we're applying, there's no way of looking at that efflux value of that. We do, in-house, we do deploy some excipients like vitamin E, TPGS, which have shown strong levels of, ability to inhibit PGP and BCRP, but we can't get that data in vitro, to my understanding. but you can see a solubility boost from those. However, we can't use that to look at PGP inhibition. Right, Bullet? Confirm if I'm wrong.
No, it is, yeah, it is correct. Our membrane is for passive transmembrane permeation.
Okay, I guess another question about the membrane. How do you check for integrity of the membrane in acceptors for a 10-hour run?
Oh, hey, Corinne. Good question. Kind of the same check that you would be doing at the end of a 2-hour run or a 4-hour run. One, you'll be able to look at your spectra, I think, of your acceptor component and be looking for any change to see if that was compromised and what else is going across. However, there's drug going across. But you can pull out the acceptor compartment when you're done and really get a visual inspection of that. You can mark a line on there where the media was filled to. You can check to see the integrity, if there was any leakage going on through there. Usually a visual check is all we do in-house, and I look at the acceptor data because it's pretty easy for me to tell if I did accidentally blow out a membrane when setting it up.
Okay, next question. How do you decide on the working concentration when working with tablets or capsules? And do you ever use multiple tablets in the same 900 ml vessel?
I'll answer the second part first. Yes, we've used multiple tablets and vessels before for these testings. Common in some cases, especially whenever we have a product like the innovator product is something we're reformulating that was maybe a 25 milligram tablet and we have a 100 milligram tablet. Assuming that the dissolution is taking place really quickly in the acidic phase. And then the first part of that question, picking the concentration, really depends. Again, it's kind of like based on some experience. You'll see a lot of papers where you find your solubility and it's some factor above that, like 2x to 5x, and that's all going to determine how quickly you precipitate down. We kind of fix somewhere between 3 to 5x that solubility to drive some precipitation without overly discriminating, because as I showed, over discrimination isn't always helpful. And so we kind of try to pick a sweet spot, but it really depends on your drug.
Okay, thank you. Next question. What level of difference, for example, 10% difference in acceptor would you feel confident with for successful bioequivalence?
It's a really good question. I think it depends. For me, when I see it, and in this case, I think the slide is a really good example. This is 3 micrograms per ml difference, and it was a 25% difference in BA. And so I think you need to keep in mind your BCS classification of your drug, your permeability of your drug. Some of these compounds that I'll see, my flux values are 100, which is very high, or even 150 in some cases. And so like seeing a value of 120 versus 110, may not be as seeing a significant, you might not even be able to see a significant difference when you go to in vivo here. And so I think it's really depends on how permeable your compound is and what these levels are here. And maybe not a strict percent difference, but just like taking into account everything you know about your compound to kind of get a gut feel of how sensitive it is or how quick the precipitation is taking place in the donor compartment.
Okay, thank you. Another question. Have you used co-amorphous systems in MacroFlux, and what's the viability to use ASD instead of co-amorphous systems in dogs?
So for the second part of that question, I'll take it as a co-amorphous system is an ASD, falls under that blanket. And so we have done co-amorphous products. We've done both where we've manufactured them separately and tested them separately, manufactured them separately and tested them together, and then manufactured them together and tested them together. as you've been to Bullen and Pion's advanced fiber optic training course or discussions or learn those tools, you can capture both of these in both the donor and the acceptor compartment and be able to differentiate these, assuming that they have, distinct UV spectrum that you're capturing in this, and it's just as equally as a tool with one drug as it is two drugs, whether it's a co-amorphous system or just two ASDs within solution. I would treat those the same because most likely the co-amorphous systems, you're just getting additional stabilization and dissolution rate enhancement from one another, but they're going to dissociate once they're in solution and permeate separately from one another, unless there's a strong interaction, and then most likely they're not going to permeate across.
Okay, thank you. I think we have time for a couple more questions. Let me see. Next one. Do we have literature comparing in vivo and flux data rank ordering for class 2 and class 4 drug, saying importance of flux than dissolution?
That's a good, I don't know if Blunt, we've done a, say we, y'all have done a strong review paper looking at just that. I know there's correlations of using, the flux and how that's correlated to both in vivo and human data over time and tested. Differentiating that down to that breakdown between a BCS class 2 and 4, you could probably dig that out. I think, Bullet, can you chime in?
Yeah,
I can give my thoughts.
yeah. I don't think. Yeah, I'm pretty sure there is no like proper review paper like summarizing that. There are quite a few case studies on BCS class 2 compounds and fewer on BCS class 4. Yeah. One is in way on class 4, but yeah, in terms of like having a review, that's maybe something we should consider to summarize all these.
Yeah, if I had to guess, there would be a lot more correlation between some of the 2 data than the 4 data due to the high variability with the 4, the susceptibility to colloidal formation. It's just a little bit more challenging, I think, to characterize, and it's what we did here, right? You can see it in this, and I wouldn't say every 4 takes place like this, but when you have a low permeability, and then whoever asked the PGP question, and then you have some efflux, it's really, you're getting into a lot of iterative development, I would say, with some PK data and doing some modeling at that point.
Okay, thank you both. I think this is all the time we have for now. I want to thank you all again for joining us today and thank our speakers for their contributions. If we didn't get to your question, we will cover it offline. Again, you will be receiving a link with that will take you to the recording of this webinar. And as always, be sure to check our website for upcoming webinars and events. Thanks again for joining us today and have a wonderful rest of the week. Goodbye for now.
Thanks, everyone.
Thanks. Goodbye.